Hello, and welcome to our rapid-fire presentation, leveraging pathophysiology and context of use to identify NASH clinical trial biomarkers from Celerion Bioanalytical Sciences. We are a global clinical research organization working with partners leading clinical trials and biomarker discovery in metabolic diseases. My name is Sumit. I lead biomarker development for clinical trials at Celerion for patient enrollment, safety, and innovator efficacy. For the past decade, biomarkers have been a hot area of research because of their potential to improve the probability a clinical trial progresses while saving time and cost from invasive trial endpoints. This will only grow as more and more preclinical and omics-level data is available, enabling precision medicine. Despite this, only a few biomarkers so far have been validated for trial use cases and for a limited number of diseases, and the chance of failure if a biomarker being translated to a trial is very low. The current workflow for translating a biomarker to a clinical trial or clinical use uses a bottom-up approach. Typically, biomarker analysis starts with in vitro or preclinical models and uses genomics or other omics-based approaches to identify potential biomarkers. Eventually, a selection of these markers is tested in an early clinical trial and further developed. But this approach has disadvantages. The preclinical models must be available and accurately represent the human condition. The use of omics identifies many biomarkers that can lead to false positives and a high failure rate. And this approach also doesn't link the biomarker to a clinical context of use, making it unlikely it'll be translatable to a trial. Today, I'll be describing an alternate reverse engineering approach, which we have found valuable for biomarker discovery. In this approach, biomarker discovery starts by defining the context of use. We want the biomarker to be used in the clinical trial. Next, we search literature and omics databases to find biological pathways and pathophysiology that link a biomarker to that desired context of use. Finally, we validate the biomarker assay and validate the biomarker clinically against established endpoints. This makes the biomarker ready for a clinical trial and FDA submission. In this approach, preclinical models may not be necessary, saving time and cost of finding an appropriate model. The biomarker is also linked to a clinical trial context of use directly, making it more rapidly translatable. In this approach, as I will demonstrate shortly, defining the context of use is critical to having a successful translatable biomarker. Shown on the left are FDA examples of biomarker context of use, particularly for clinical trials. These definitions will help you determine which biological pathways to investigate for biomarkers. After possible biomarkers are identified, it will also help you determine which analytical tests are needed to validate the biomarker assay and the clinical endpoint the biomarker should be compared to. Both of these components are needed by the FDA when using a biomarker for a trial. I'll now present a case study where we used this approach and found success in a translatable biomarker. Our case study is from a study where we were searching for biomarkers for use in non-alcoholic steatohepatitis or NASH clinical trials. Our specific context of use was to find a biomarker of patient selection, inclusion-exclusion criteria, or a prognostic enrichment of NASH progression, and thus appropriate for inclusion in NASH drug development clinical trials. We then used clinical and preclinical published literature as our data source to find candidate biomarkers. We then measured the biomarkers and did a validation of the biomarker assay and compared the biomarkers to clinical endpoints. In our case study, the endpoint was a comparison to the FibroScan transient elastography ultrasound during patient enrollment. So we searched literature and identified the biological pathways in NASH to identify biomarkers. Shown here is the pathogenesis and possible biomarkers for each stage of NASH involving steatohepatitis, inflammation, cell death, and finally fibrosis of the liver. Our context of use was a prognostic and screening biomarker. This suggested we needed a biomarker involved in the middle of the pathogenesis and not a later stage biomarker like fibrosis markers. We chose the biomarkers of cell death and apoptosis. Specifically, we measured caspase-cleaved CK18. So further on this biological pathway, CK18 is an apoptosis biomarker. It's from a keratin fragment present in epithelial cells, and the K18 form is specific to the liver after it is formed from caspase cleavage. This suggested that it would be a specific marker for liver cell death and be appropriate for trial inclusion-exclusion biomarkers. The literature also indicated cell death occurs in the middle of the NASH pathogenesis, making it a good candidate for a context of use as a prognostic or screening biomarker because elevations would occur earlier than fibrosis biomarkers would. The context of use also drives the analytical technology and assay design. We wanted a biomarker specific to apoptosis that is specific to NASH, as opposed to a CK18 release from necrosis from alcoholic liver diseases. That's why we chose M30 apoptosense ELISA that measures the CK18 fragment that is only formed during apoptosis. The next step after a biomarker is chosen is to analytically and clinically validate the biomarker of interest. This is an example of fit-for-purpose assay validation tests that need to be done depending on the context of use of the assay. As a biomarker is more and more critical to the trial, more tests are needed to validate that the assay is performing well. In our context of use as a prognostic biomarker, we chose a Tier 2 validation and performed the necessary analytical tests of the CK18M30 assay. The tests we performed are to determine the precision and accuracy of the test, first of all. We also had an endogenous quality control for the serum of NASH patients to monitor the assay over time. We also performed stability tests and tests such as parallelism, which confirm that the recombinant standard used in the assay for a standard curve is comparable to CK18 found in the body. This is necessary for a Tier 2 partial validation. For the clinical validation of the biomarker, we compared the biomarker concentrations to an endpoint that fit our context of use. In this case, we used a test commonly used during pre-screening of NASH patients during trials, which is our context of use. This is the FibroScan transient elastography that is based on ultrasound and can quickly and non-invasively measure liver steatosis, making it easy for pre-screening patients. We screened and measured 200 patients with the FibroScan for liver steatosis and measured CK18 M30 concentrations in their blood. We found that CK18 dramatically increased with increasing liver steatosis, shown here as a higher cap value from the FibroScan. This shows that our reverse engineering approach is identified CK18 as a possible biomarker for pre-screening patients in a NASH clinical trial. The reason defining a context of use is important is because it helps you know which biological pathways to target for biomarkers. When the appropriate pathway is not chosen, the biomarker will not fit the context of use when it is compared to the clinical endpoint. For example, in our case study, we did a separate study with a different context of use. In this case, the context of use is to find secondary endpoints for monitoring and staging liver fibrosis during clinical trial. In this case, CK18 did not increase with increasing liver fibrosis, determined clinically by a liver biopsy. This demonstrates that cell death and CK18 should not be used as a biomarker for fibrosis context of use. Instead, we found other markers that correlated with fibrosis and are likely involved with fibrosis pathophysiology. Another benefit of this approach is that because clinical data is derived early without preclinical models, it can also be used to derive mechanistic insights that can help you with therapeutic identification. For example, in our case study, knowing that CK18 is elevated during human steatosis, but not correlated with fibrosis, allows us to understand when apoptotic cell death occurs during the progression of NASH, and may be helpful information for therapeutics wanting to target cell death. In summary, this reverse engineering approach to biomarker identification can help identify a biomarker without using preclinical models. In our case, we used biological pathways that were published, but omics databases can also be used. The link of the biomarker to a context of use and comparison to a clinical endpoint really increases the chance that the biomarker can be used for a clinical trial. Another of this approach is that if a biological pathway is shared with your context of use with another disease, it can be quickly translated to a different disease without starting from scratch with another preclinical model. For example, liver fibrosis and lung fibrosis share common pathways and may share biomarkers, but just need a different clinical endpoint comparison. I'd like to thank those involved in this work, the Diapharma and VLV scientists who assisted with the M30 apoptosense kit, and Natalie and Sabina from Celerion who helped with the analytical and clinical validation. Thank you for watching our presentation, and please contact us with any questions. Thank you.